Cool. Good morning, everyone. Thank you so much for the introduction. Yeah, I think that after what we heard about personalization and how precision medicine is important, I'm very happy to present this presentation soon after Segun Fatumo. So basically, I'm part of the Human Nutrition Unit, which is this unit in Parma, Italy, in which we are investigating how nutrition and how, of course, inter-individual variability can help us in understanding how what we eat and how the environment where we live and how our genomics and the omics that we can measure can actually help us understanding more about personalized nutritional strategies and, of course, precision medicine approaches. So I'm quite new to the world of polyphenols. That is our main focus. Basically, polyphenols are these bioactive compounds that are present in all plant-based food. And it's just metabolites that have one or more phenolic group attached to them. There are different classes, and basically you can see all the wide variety that is present in nature and basically in all of what we eat. And basically these bioactive are important because they trigger health, they have health promoting effects, particularly regarding cardiometabolic diseases. And this is actually our focus. And when I started investigating 2 years ago these metabolites and their effect on human genomics and human omics, I started with a question that was like how we can understand about something more about the genetics of these metabolites. And of course, if we have a reference to refer to when doing Nutri-omics and multi-omics investigation. So we started assessing in an inferential and functional way whether some of the genetic classes that were interacting by some studies that were reported in the literature, if they had an effect on the polyphenols, and of course, what was the availability of information in the biobanks. And basically performing some network analysis starting from a plethora of functional classes such as UDP, glucuronosyl transferases, ABC transporters, sulfatases, and sulfotransferases, how we actually, we found out that they were actually strictly connected and related one to the other. The string network represent basically the biological complexity. And of course, provided us more information regarding what was the pathway, what was the information publicly available on data sets. And as we find here was actually basically that we find some cluster mostly related to the functions that are happening and basically biological pathways related to polyphenols, such as glucuronidation, xenobiotic transport, and so on, sulfation and other functions. So looking at that, we were actually saying, okay, we do have some information coming from genomics and coming from other studies, so we can move on to our studies to actually try to understand in this ERC-funded project, the Predict Care project, how these metabolites can actually trigger health effect and how is their effect on human individuals. Basically, this was led mostly by the PI of this study, Professor Pedro Mena at the University of Parma, and was the PhD project work of my colleague, Cristiana Mignogna. Basically, we had this cohort of 300 individuals that we characterized at a different level, from genomics, from urine samples, from blood samples, basically doing some metabolomics to have a lot of information regarding metabolites and the samples. And then we try to deep phenotype and characterize several omic layers, all the information that we can retrieve from questionnaires, from genomics, from other omics, and of course from metabolomics and the microbiome, which are the major focus when studying polyphenols and doing personalized nutrition study. This was actually our core. You can find here some stats. And basically, we enrolled for 413 individuals from the Parma area, so basically in the north central part of Italy, and just 297 were the one completing these studies, which consisted of a diet of an acute supplementation of polyphenols to see how polyphenols can actually have an effect. These were all the information that we collected, basically anthropometric measurement, nutrients, metabolites, cardiometabolic markers to assess cardiometabolic health, and of course, blood markers, stool and urine, blood samples, stool or urine for microbiome, metabolites, and of course, genotyping and biomarkers. The nutritional field is basically revolved around metagenomics and metabolomics. I will start with metabolomics, and this was the work by a lot of colleagues in which they identified and quantified a lot of metabolites, a lot of phenolic metabolites. 208 of them were retained and were actually classified, and we used for further analysis. And these analyses didn't actually focus more at the moment on the metabolite levels. Rather than we try to pull together these metabolites into metabotypes, meaning that phenolic and phenolic metabotypes, so phenotypes based on the metabolic signature of this compound. This was achieved by this sort of model in which we tried to investigate more than 1,500 models and then performing a lot of transformation, scaling, dimensionality reduction, and clustering. This combination was actually the one performing at best for our studies. And we assessed this some sort of cluster, which we define quality-quantitative cluster, which is quite common in nutritional investigation, in which we identified two, I'm not saying separate, but two different groups, in which the blue one was the metabotype 1 having a low phenolic excretion, and the one in green, metabotype 2, having a high phenolic excretion and production. And we try to assess and investigate the contribution of the two metabotypes in terms of cardiometabolic health. And we actually try to assess and investigate whether low phenolic production was associated with a high cardiometabolic risk and vice versa for the metabotype 2. So going after that, as I was saying, the vast majority of nutritional studies revolve around metabolomics and, of course, metagenomics. So we did a standard workflow in which we use ITS sequencing to understand the microbial composition of the fecal samples from our individuals, and we find this some sort of 100 families, 258 genera, and 483 species. However, when trying to understand the microbial environment and the microbial community, we didn't find any significant differences in alpha and beta diversity across our samples. And however, when trying to investigate in some sort of fold change-based analysis, we find that particularly in the green part, some of the species where I had a different fold change compared between the two metabotypes, and some of these species were actually related more on the production and the excretion of polyphenols, such as flavonifactor plauzi and pseudoflavonifactor plauzi. And in the end, starting my work, basically, I'm a human genomicist by education and training. And we started working on the genomics, and basically, we performed this kind of workload, genotyping, while the human GSA, the Illumina GSA genotyping kit. We performed genotype imputation, then we moved to genome-wide association studies and computational functional analysis on something like 6 million variants across the 300 individuals, divided by 128 from metabotype 1, having low phenolic production and high risk for cardiometabolic diseases, and the 169 from high risk, high phenolic production and low cardiometabolic diseases. This was actually, to our knowledge, the first genome-wide association studies that is currently under revision and publication. And having some sort of low sample size, we decrease our genome-wide significant threshold and suggested significant threshold to 5 and 3.5. And, And basically, performing some functional analysis and understanding with FUMA-GWAS, we find out that we identified 3 different genomic risk loci. FUMA-GWAS is limited to 10 to the minus 5 as a significant threshold. And in these 3 genomic risk loci, the vast majority of variants was actually intronic and intergenic, meaning that we were not seeing any exonic difference translated to the protein function in our results. Of note, the first genomic risk loci in chromosome 14 was the one having just a gene physically located in this risk loci. And here we have some sort of Manhattan plot for the chromosome 14, in which we identified association for these genes that was never reported being of interest for polyphenols and polyphenols metabolisms, which was the thyroid stimulating hormone receptor. However, doing some research and some experiment, we find out that he has affinity binding with phenolic structure, being actually a receptor for hormone structure that shares some similarity with our metabolites. So we are further investigating on that, and we then also look for our genes of interest, and we find out that some of them, likely for the small sample size that we analyzed, or also because of they were not too much significant, we are trying to assess that with other studies. And others of the gene of interest that were involved in polyphenols metabolism were actually significant. And of note, we find out some genetic sets which was actually possibly related to positional and regional effects in terms of epigenomic and transcriptomics that, however, we cannot investigate furthermore. And since the polyphenols are thought to have an effect on cardiometabolic health, we tried to assess using some sort of polygenic risk score correlation from the polygenic score catalog, whether they can actually improve our understanding on the cardiometabolic effects. This was mostly because of when tried to assess the cardiometabolic effect of polyphenols in correlation with cardiometabolic biomarkers, we didn't have any actually information and significant correlation between the status of belonging to one of the two metabotypes and actually the biomarkers that were available and the risk scores that were available for all the samples. So we tried to search into the polygenic score catalog. We extracted all the scores, applied them using the PGSC calc Nextflow pipeline that is developed, and basically doing some statistical analysis to see if already computed polygenic risk score can actually help us understanding more on the cardiometabolic health of our samples based on the two metabotypes. We computed these scores, we did this pairwise t-test with non-Ferroni correction, and actually we retrieved 180 significant polygenic risk scores that we try to group into classes, and as you can see here is actually the grouping in which we focus more on cardiometabolic health, proteins that are related to cardiometabolic diseases such as lipoproteins and so on. And this is just an example in which we find some differential significant between the scores of the two metabotypes. In red, the metabotype 1, so the one higher risk, and in blue, the metabotype 2, the one at lower risk. And actually, we are now trying to understand whether this inferred genetic cardiometabolic health can be of help to understanding more about these metabolites and their function in our organisms. And of course, of a possible influence of the genetic background. As we were saying, these are mostly European individuals, but not just actually, we do not just have European individuals in our cohort. So likely, this represent basically the genomic complexity and the omics complexity that we are now trying to assess for investigating metabolomics and, of course, metabolite related to cardiometabolic health. So in the very end, as I think also this afternoon in which we're having this talk on multi-omics, we are now trying to integrate, and I'm not presenting this data so far, but we are currently trying to understand, basically using Mixomics, Diablo, but also other tools, if there are any correlation on all these single omics analysis that we performed so far. You have a B-plot on the right side, on your left side, and basically a circle plot trying to correlate all the different metabolites, metagenomic species, and genomics variant that are present, and try to understand if there is any correlation actually, not just for the single omics, but for the pool and joint contribution of all the omics together. And if you want to know more about that, the PhD student of our group, my PhD student Lucia, is actually presenting today a poster and on Tuesday on the Transmed session. So you can ask her about this analysis that she performed so far. Yeah, basically concluding this kind of overview on this project, we identified these metabotypes, so metabolic signature pool phenotypes, in which we identified basically two clusters that might be related to cardiometabolic health. However, we are now trying to understand the levels of the metabolites and how the levels of every single metabolite of these 208 metabolites can actually influence on the cardiometabolic health and can have healthier and health-promoting effects on that. The microbiome didn't show very significant results. However, when doing some other analysis, we see that some species that are key to polyphenols, metabolisms, and bioavailability are actually trying to differentiate between the two metabotypes. We are now moving into genome shotgun metagenomics sequencing to have the whole picture depicted. Regarding genomics, we did not find that so-called genome-wide significant signals. Actually, they were more related to changing the threshold and try to understand for the very first time the genomics analysis and the genomics behind all these metabolisms. And we are now trying to pull together everything and to delve into the unknown. Basically, in the nutritional field, these are kind of new analyses that we are performing so far. So we are now trying to understand the non-coding region contribution, the intronic contribution, and intergenic contribution. And of course, all these omics pulled together to try to understand how we can explain with multiple omics just what we are normally doing. So basically eating and try to survive, and how eating actually has an effect on our health. Basically, this is what we are doing so far. So try to collect as many omics as possible, try to pull them together, and then try to give precise recommendation basically for personalized nutritional plan, and of course to personalized medicine approaches. And we are now moving from the healthy individuals of the obesity project to these two new projects, one called the Perker Diet. I'll try to investigate the effect of these metabolites in people being at risk of cardiometabolic health. And of course, on the mentoring for health, so trying to understand this effect on non-communicable diseases and lung cancer patients to try to assess whether they have also not just a health promoting effect regarding cardiometabolic diseases, but also a health promoting effect on cancer patients and on more complex diseases. Of course, this is not just my work. This is basically all the group and all the people that are contributing to the realization of this project. And I want to thank, starting from the PI, Professor Pedro Mena, but all the other components. And of course, I want to thank you all for your attention. And I'm very happy to welcome any of your questions.
Thank you for a great talk. And we have time for probably about one question, if anyone has any questions. Okay. Well, thank you.